So this is a Scribble video that we'd love you to listen to about a paper we've recently published in Cell Reports. The title of the paper is Proteolytic and Opportunistic Breaching of the Basement Membrane Zone by Immune Cells During Tumour Initiation. And it's a collaborative effort between two labs actually, mine in Bristol, I'm Paul Martin, and Yi Feng's lab in Edinburgh. So it's known that when a cancer within the epithelium grows big enough, in order to become malignant, to metastasize, it has to breach the basement membrane, which is an extracellular layer beneath it, in order to invade the underlying connective tissue. And that metastatic spread is when the prognosis for the patient becomes considerably worse. We are interested in a much earlier episode during tumour initiation stage, very early stages in tumour initiation, when there's another breach. This time it's of immune cells moving in the other direction to visit the cancer clone. To look at these very early cancer initiation stages, we use zebrafish larvae as our model, because you can see right through, it's translucent. Zebrafish larvae are also genetically tractable, which means we can fluorescently tag in complementary colours our cancer cells and our immune cells and look at their dynamic interactions in real time. So if we drive in any particular cell lineage within the skin, a GFP and a human mutant oncogene, we can watch from the moment they're born these individual cells expanding and growing, becoming a preneoplastic cancer clone, and we can live image how the red immune cells, neutrophils or macrophages, interact with them. What's clear from studies we've shown already is that those innate immune cells, both neutrophils and macrophages, deliver trophic signals to the preneoplastic clones that are required for them to grow. In fact, if you knock out the innate immune cell lineages, then those clones don't grow.
A transverse section through a clone shows the cancer cells clustered together in the skin, sitting on the basement membrane. The inflammatory cells are found in the tissue beneath the basement membrane, so in order to access the skin cancer cells, they must breach this barrier. To study this basement membrane breaching, we use several transgenic zebrafish. First up are our skin cancer models. Larval zebrafish skin is very simple, with three main cell types. Mucus-secreting goblet cells, which are embedded in a bilayer of epithelial cells. Sitting directly on the basement membrane is a layer of basal cells, and on the skin surface, a layer of superficial cells with their distinct microridges. We're able to drive each one of these lineages to become early cancer cells by expressing a fluorescently labelled active form of the human oncogene RAS, called HRAS G12V. Directly beneath, and closely and continuously associated with the basement membrane, is a distinct layer of collagen 1. Recently in the lab, we have developed a transgenic fish where this molecule is labelled with GFP. We call the collagen 1 layer and the basement membrane together the basement membrane zone, and the collagen 1 GFP fish has allowed us to use the collagen 1 layer as a proxy to live image events at this extracellular matrix barrier that separates the skin from the dermis below. From transmission electron microscope studies, we show that immune cells access cancer cells in the skin through holes or breaches through the entire basement membrane zone, and looking closely at these micrographs, it appears the immune cells can make these holes by enzymatic proteolytic events. Proteolysis, however, is not rate limiting for cancer growth, as when we count the number of immune cells accessing and making contacts with skin cancer cells, they remain unaffected when the experiments are done in the presence of protease inhibitors. Further live imaging, combining our collagen 1 GFP, skin cancer and labelled immune cell transgenic zebrafish, show us that immune cells migrate along the dermal underside of the collagen 1 layer, and they're able to access skin cancer cells through naturally occurring holes in the basement membrane zone that are found in particular along a region called the horizontal myoseptum or midline. These holes provide routes of least resistance for immune cell migration, and we describe them as portals. We show that cancer cells growing in the vicinity of these portals in the midline receive more immune cell visits, and they grow more than clones growing further away, indicating a rate-limiting role for the basement membrane barrier at these very early preclinical stages of tumour development.
Our lab is a wound healing lab. A little while ago we published a study where we showed that if you superimpose on this system a wound, you get an acute inflammatory response, and the neutrophils and macrophages drawn to that wound are subsequently drawn to preneoplastic clones in the vicinity and encourage their growth. Now we know from this study that as well as the wound releasing damage attractants, drawing immune cells, the wound itself makes a vast breach in the basement membrane that'll act as a portal, enabling immune cells access to the epithelium in which the cancer cells are sitting. So what's the clinical relevance of what we've described to you? We're wondering whether these weak spots in the basement membrane, these portals as we describe them, might reflect the microperforations that are seen in clinical materials within, for example, the gut and airway, that associate with inflammatory pathologies, which in turn associate with cancer progression. And so these microperforations, these portals, may be biomarkers for cancer progression. They may also be potential therapeutic targets for intervention. Thank you for listening to our Scribble video.